A patient walks out of a manual therapy clinic. Their restricted range of motion is gone. The joint feels loose. But within hours, a deep, localized soreness sets in, right where the therapist applied deep shearing pressure. This chart graphs the clinical paradox. Within 5 minutes of friction, tissue stiffness drops to 0. A secondary wave of soreness peaks at 12 hours, resolving near 48 hours. We assumed this was physical micro-tearing from the therapist's hands. The data tells a different story. This 12-hour soreness is actually the manifestation of a highly specific, mechanically catalyzed inflammatory cascade. Misinterpreting this paradox is a massive clinical vulnerability. When practitioners and patients assume this localized pain is a negative side effect, they often intervene to suppress the exact biochemical mechanism required for long-term tissue restoration. The entire timeline of this inflammatory cascade is controlled by a single master regulator. It is a glycosaminoglycan known as hyaluronan, or HA. Throughout human vertebrate tissue, HA functions as a vital space filler and a high-capacity water-binding lubricant. In manual therapy, its most critical location is within the extracellular matrix, the liquid environment sandwiched precisely between the gliding layers of deep fascia. The defining physical variable of HA is its molecular weight. Because it is a dynamic polymer, the physical size and length of an HA chain strictly dictate the biological signals it sends to surrounding cells. Tracking the degradation and mass of this single molecule reveals the exact mechanism determining if a patient feels stiff, if they feel acutely sore, or are completely cured. Under physiological conditions, healthy HA exists as high molecular weight HA. These are massive, entangled polymer chains sitting comfortably above 1,800 kilodaltons. At this enormous size, HMWHA utilizes its negative electrical charge to retain vast amounts of water. This creates a highly fluid, frictionless cushion, allowing adjacent fascial layers to slide smoothly past one another during movement. When homeostasis in the extracellular matrix is disrupted, this sliding mechanism fails. Clinically, we call this failure densification. Densification is a specific structural shift. High molecular weight HA heavily self-aggregates and cross-links with other proteins. As chains bundle tightly, they saturate their water-binding links. Local free water plummets. Without water, the matrix transitions into a rigid, viscous gel superstructure. The fascial layers lose their ability to glide. As the patient tries to move, this dry, immobilized tissue physically pulls on adjacent free nerve endings, generating a constant, dull pain. Chronic musculoskeletal stiffness is a localized fluid viscosity crisis. Deep friction manual therapy applies concentrated mechanical stress directly into this rigid gel. Pressure creates friction, generating localized heat. For the intervention to work, the matrix temperature must surpass a precise thermodynamic threshold, 40 degrees Celsius. At 40 degrees, exothermic heat combines with mechanical shear force from the therapist's hands. This dual attack disrupts the weak van der Waals forces holding the HA superstructure together. The rigid gel breaks down, viscosity plummets, and fascial gliding is instantly restored. The joint moves freely again. But the excess, aggregated HA chains haven't vanished. They are still physically trapped inside the fascial compartment. To clear them out, the body relies on depolymerization. The intense physical shear stress from the manual therapy literally fractures the native HA chains into smaller and smaller pieces. As the physical length of the chain snaps, its molecular weight drops. The fragmented HA now has a completely different mass than its parent molecule. Breaking the physical stiffness solves the immediate mechanical problem, but fracturing those chains permanently arms a secondary chemical time bomb within the tissue. Depolymerization produces low molecular weight HA, fragments measuring 250 kilodaltons or smaller. At this size, HA's biological function flips. Unlike its inert parent, LMWHA is highly pro-inflammatory. These fragments interact directly with cell membrane receptors, docking into the CD44 and TLR4 pathways. Once docked, they trigger a massive activation of the NF-κB pathway inside the cell. The cell responds by pumping out inflammatory mediators, including cytokines like interleukin-1 beta and destructive reactive oxygen species. This creates a rapid positive feedback loop. The inflammatory mediators recruit destructive enzymes like hyaluronidase, which chop up even more native HA into low molecular weight fragments, amplifying the chemical signal. This localized chemical storm is the precise biological source of the patient's soreness. As the fragments accumulate, the pain climbs, hitting its peak right at the 12 to 24-hour mark. The patient's next-day pain is the exact chemical marker confirming that the mechanical depolymerization of the stiff tissue was successful. The cascade is designed to resolve itself. The breakdown process continues, driven by cellular enzymes like Hyal-2 and Hyal-1. These enzymes force the fragments into the final stage of degradation, oligosaccharides. These are the smallest possible HA pieces, weighing less than 10 kilodaltons. Here, the biology pivots one last time. These tiny oligosaccharides act as an alarm off switch for the entire inflammatory pathway. They possess potent anti-inflammatory properties. They actively downregulate the release of cytokines, suppress cellular apoptosis, and stimulate fibroblast motility to repair the matrix. Because they are so small, these final fragments can easily drain out of the local tissue via the lymphatic system to be cleared by the liver. Clearing the compartment of broken HA removes the blockage. This triggers local cells, called fasciocytes, to synthesize fresh, healthy, high molecular weight HA, permanently restoring tissue homeostasis. Look at this symptom graph again. The drop in stiffness aligns perfectly with the breakdown of high molecular weight HA. The delayed soreness peak maps exactly to the appearance of low molecular weight HA, and the final pain resolution mirrors the arrival of the oligosaccharides. Physical friction, heat, and mechanical shear stress directly dictate the timeline of biochemical depolymerization. The resulting shift from pro-inflammatory LMW fragments to anti-inflammatory oligosaccharides dictates the timeline of the patient's pain. Effective manual therapy is an act of mechanical to chemical signaling, forcing a stagnant biological matrix to finally turn over. Post-treatment inflammation is a pro-defensive requirement for tissue remodeling. If a patient takes nonsteroidal anti-inflammatory drugs to suppress the soreness, they artificially halt the cascade mid-cycle. The breakdown stops at the painful low molecular weight stage, and the tissue cannot clear the compartment to rebuild. If analgesia is absolutely required to tolerate the 12-hour peak, paracetamol provides pain relief without physically blocking the critical HA fragmentation pathway. To permanently fix a mechanical joint or tissue issue, practitioners and patients must possess the restraint to let the biochemistry run its full course.